What do you know?
Well, good afternoon, everyone. We're waiting for a few more people to join us. Good afternoon.
Hey, Valerie, how are you?
Good. How are you?
Doing well, thanks. Good to see you.
Good to see you as well. We'll give everyone a chance to join before we get started.
Hey, Mark, it's Jay Mason. Nice to see you.
Hey, Jay, how are you?
I'm well, thanks.
You can't even spell your own last name.
Is it on there wrong? Oh, well.
And I am not Alessandra. I am Fabio.
No, you're not.
I'm using my wife's computer. Apologies.
Is that Fabio? Hey, Fabio.
Yeah.
Hey Fabio, hey Jay.
Hi.
So my audio might be a little bit iffy because apparently an alarm drill just went off on my building. So I hope you can hear me well.
Well, that's exactly right. So you may hear some background noise. We'll give everyone another minute before we get started.
Hey, Val, this is Amita. So nice to see you.
Good to see you as well. We're just giving everyone another minute before we get started. We'll probably start about 2 or 3 minutes after the hour. We will let you know, we do want you to know that this is being recorded and it will be on our social media sites as well as our website.
Hi, Valerie. Nice to see you. This is Tayo.
Hello, how are you?
Long time.
Good.
Doing well.
I know. I'm still here.
Well, I left FGA about 4 years ago.
Wow. It's great. Yes, it's great that so many of you are still in this space. So it is good to see a lot of very old friends. Not old. I have to take that back.
Always young, right?
That's right. That's exactly right. Well, I think we're going to get started in the interest of time because we actually have a whole lot to cover today. I'm very excited about this webinar that we have the opportunity to host. We meaning the CSRC. For those of you who are joining and haven't worked with us before, the CSRC is a public-private partnership that originated back in 2006 with the between the FDA and Duke University. Our focus has been on cardiac safety issues related to drug and device development. And as part of our initiative, we have always had an ECG warehouse. And today we will be focusing on changes that are actually impacting the ECG FDA warehouse. Many of you have maybe already received notification about some of the changes, but we thought this would be a great opportunity for our members to have a firsthand discussion with Dr. Jose Vicente about the changes that are occurring. For those of you, I just want you to know that if you have any questions or comments, we want you to use the chat box and post your questions. Also, as you probably have seen on the flyer, Dr. Jonathan Seltzer was supposed to be the moderator, but he is unavailable, so I'll be your moderator today. And we are going to follow the agenda that we have discussed earlier. We're going to start off with Dr. Jose Vicente from the FDA. After that, we will have Dr. Cindy Green, who is from Duke University, who's a biostatistician, talking about the ECG warehouse at Duke within the CSRC. And then finally, we'll have Dr. Salim Idris, who actually is working with the CSRC on a pediatric ECG warehouse related to sudden death in the youth project. I won't take up any more of your time. I'm so thankful that each and every one of you have joined. And I am looking forward to the conversation that will happen after the presentations. Now I'm going to turn it over to Dr. Vicente, and we will move forward from there.
Thanks,
Okay,
Valerie.
you're on
I was on mute.
Yeah, there you go.
Okay, so you can see my screen right now, right?
Yes, we see it.
Yes,
Okay.
you're good.
Thanks, Jay. Okay, well, thank you everyone for calling in today. Thanks to the CSRC, Valerie, and Saimia for organizing this and give me this opportunity to give a brief overview of the different changes that are ongoing at FDA related to the ECG warehouse. My name is Jose Vicente. I am a senior staff fellow in the Division of Cardiology and Nephrology, which used to be cardio-renal for those of you who have been long enough in the space. And one of the first things I want to say before getting to even the agenda is that what is changing and what is not changing, and I will reiterate this later on. So there are no changes on the ECG requirements from the agency. That's the first thing I want to say. And the second thing is that what is changing, and the only thing that is changing is how we will receive annotated ECG waveforms. And that is changing from the ecgwarehouse.com website to receive directly through the electronic submission gateway that the agency has in place already for a few years. And the data will be the same. So it's still HL7, AECG, or the so-called XML files that we expect that you have been submitted in the past, and that we keep that as our backend. So let me move into the agenda. So I'm going to start with a brief history on how we got here, and then I will jump into what is the role and the value of the digital annotated ECG waveforms at FDA. In other words, how we use them. And the second part, really not the third, but it's the third bullet, but it's really the second part of the presentation is more about how the ECG waveforms flow from the clinical trial or the sponsors all the way to the interdisciplinary review team, which is the team responsible for reviewing studies conducted to assess proarrhythmic potential or QT prolongation potential of in clinical studies in ICH14 pretty much. So what is the ECG warehouse? The ECG warehouse is part of the technical response. It's within a more elaborated response to unexpected cases of drug-induced QT prolongation and torsade de pointes that happened in the late 1990s and early 2000s. And Cindy will cover this later on, so I'm not gonna just comment on what a normal ECG versus a torsade event or QT prolongation is. And what I wanna tell you is that there is this large database of electrocardiograms, digital electrocardiograms. And these are ECGs that are collected in clinical studies, typically to fulfill the ICH14 guidance requirement, which is the guidance that describes the clinical study or the therapeutic study to assess potential of any new drug to prolong the QT interval or to cause torsade. And this database was possible thanks in part to the community effort in developing a data standard, the HL7 annotated ECG XML or AECG XML standard, which was not, there was not an open data format back at the time that the guidance were put in place and that the FDA launched the digital ECG initiative. And this standard has become the most widely adopted ECG standard in the world. So the warehouse also enables the agency to verify QT assessments. And in part, thanks to receiving the waveforms, we can say that the warehouse has contributed to the success response of the ICH14 approach, which basically is that no new marketed drugs have been removed from the market because unexpected QT prolongation or torsade. And in addition, so it's the verification of QT assessment is a very important point, but at the same time, this large and comprehensive data of drug effects on the ECG has allowed us to also advance in the cardiac safety field. And as of January or probably February, I don't remember not the specific month, it's March 1st, we had more than 12 million 10-second ECGs submitted to the warehouse and some continuous Holter recordings as well. So how was the ECG warehouse established? basically, in 2001, the FDA launched the Digital ECG initiative. The HL7 ECG standard was developed by the community in the subsequent years. First warehouse implementation was available in 2004. And by the time that the guidelines were adopted in 2005, the ECG warehouse was up and running. And since 2006, first through ACRADA, later through contract and different mechanisms, wafers have been sent to the FDA through the ecgwarehouse.com website. And that's been the case as of today. And these are the changes that are coming up later this month and through the rest of the year. So basically, in parallel with the requirements of receiving the clinical data through electronic submission gateway, we're just shifting the submission mechanism for the ECG waveform. So when ECG waveforms are uploaded to the warehouse, instead of using the ecgwarehouse.com website, we are requesting sponsors and applicants to just upload the waveforms together with the rest of the clinical data. And that's what's happening on March. So what do we do at the agency with these waveforms? So how we look at them? And this is one question we typically get as well. So first, let's take a look at what is the data that we have in the warehouse. These are typically digital annotated ECG waveforms that are acquired in clinical trials and that are uploaded as HL7 ECG files to www.ecgwarehouse.com or the FDA Electronic Submissions Gateway. These are typically, or most of the data is 10 seconds, 12-lead ECG extractions at multiple time points. But as I mentioned before, we also have continuous halters. And the interval annotations are usually overread by cardiologists blinded to treatment and time. Of note, all the rest of the clinical data, treatment information, some of the subject listings, drug concentration, clinical study report, clinical study protocol, ECG analysis plan, statistical analysis plan, all these things come already through the electronic submission gateways. So the reception of the waveforms is kind of the remaining part to use this single entry point. What do we do with these waveforms? So as I mentioned before, the first step is verification, or one of the main things that we do is verification of QT assessments. And the interdisciplinary review team for cardiac safety studies, which you formerly known by QT-IRT, is the team responsible for reviewing data collected and submitted to fulfill ICH 14 requirements. And as an example, these are pretty much thorough QT studies, but also substitutes of QT studies. And the analysis that we conduct in IRT to support the review include verification of raw data, for example, right? So how many ECGs were specified to be collected by protocol or in the ECG analysis plan, and how many ECG waveforms do we have? How the ECGs are annotated, how does the QT interval in the waveforms look like compared to what's in the tabulated data sets? But we also check quality signal of the waveforms, and if needed, we also assess what is called QT measurement bias assessment. And I'll get into that later on. You can check more about cardiac safety studies and how we look at them in the interdisciplinary review team website, and I'll include the link in this slide, but also in some other slides. So let's take a look at how we look at the number of ECGs and how they look like and things like that. So basically, when either through the ecgwarehouse.com or through the gateway, an applicant uploads waveforms, some of the information that is also requested is what is the application number, what is the study ID, but also whether the annotations were done in the rhythm or in the derived median beat, in lead 2, B5, what was the primary lead for annotations, number of subjects in the trial, number of ECGs per subject, total number of ECGs that were included in the upload, and how many bits were used to annotate the QT. And that's something that you can see here. So in the screenshot here, I'm showing an example using data from the so-called SIPA ECG clinical trial. In this case, from the parallel part, where there are 4,135 ECGs and that were collected from 50 subjects. And here we are seeing that there are some small discrepancies, and this is because in this particular case, there were 2 subjects that dropped out during the trial. And we can spot that pretty quickly in this 4% of the 50 here. And also that we also see that some of the ECGs don't include QT annotations. And if we look at these are very noisy ECGs. So it's not that the information is missing, it's that it cannot be acquired. If needed, we can go and take a look at either the 10 seconds strips or the derived bit, at the annotations, and check whether one particular ECG is properly annotated. Of course, the goal is not to reproduce the annotations or to manually adjudicate the adjudications, but basically, if there is an outlier, can we check this on screen and verify that it's not a spurious measurement? And as I mentioned before, we also can actually cross-reference with the raw data sets. And basically, one of the things that our tool does is take the interval annotations, either from the 10 seconds or the median beat, and then produce something that resembles what is called an EG data set in SDTM. And basically, we can get a value for PR, QRS, QT, or other intervals that are annotated in the waveforms per ECG or on the individual bits basis if needed. And that way we can cross-check together with either the file ID or the file name or the EGREF ID that the annotations provided match the data sets. In few cases, we noticed that there were some discrepancies on being the overread measurements being provided in the data sets, but the automatic measurements provided in the warehouse, or the other way around, but that can be easily solved with requesting the adequate information back to the applicant. The other part that we do is verify the signal quality of the waveforms. And here I'm showing an example for high frequency noise. And instead of having fixed cutoffs, what we do is we compare the values of a given quality metric to the distribution of the metric across all the studies in the warehouse. And in these two panels here, the wide error bar with the distributions, right, is showing the high frequency noise distribution across the one particular study, and it's the same study. While the green, blue, and orange box plots show the 5%, 50 percentile, and 95 percentile of the distribution in the warehouse. So basically what we want to compare is whether the new study, whether or not it's within the tail distributions across the warehouse. And in this case, we saw that 6.9% of the measurements of the high frequency noise were outside the range of the warehouse. And by worse here, we mean that they were outside the range. It doesn't mean that they are better or worse, just that they are off. And what we noticed in this particular study is that absolutely all the ECG waveforms, including uploaded to the warehouse, were not really the ones that made it to the primary QT assessment. In other words, they were screening ECGs that, for example, are not part of the primary analysis. There are safety ECGs that might not be part of the primary QT assessment. So when looking at the data that actually made it to the data sets for the primary QT assessment, what we see here is the distribution becomes a little bit tighter. And in this case, 4.6% of the data is outside the range. The third piece that we do sometimes, especially since the ICH 14 release, Q&A release 3 on December 2015, is to take a more careful look at the studies, whether or not they have bias in the QT measurements. And in particular, I'm referring to single and like substitutes for QT studies where you don't have a positive control included in the trial. And to do this, basically what we look at is at the difference between the QT values provided in the waveforms and the data sets compared to the QTs obtained by an automatic algorithm, fully automatic. And again, it's known that there are differences, or there can be differences between either the slope or some of the measurements between different QT measurements when using different algorithms. The goal here is to have a contrast to have a better reassurance that the measurements seem okay and that there is no bias. So let's take a look at this particular example, and this is from a public review that is available at the FDA website, where in this particular case, there were two studies, one of them for the same drug. One of them was a single ascending dose study, another one was a thorough QT study, and there was a difference in the concentration QT relationship between the two studies. So what we did to investigate this discrepancy was to perform this QT bias assessment, which is described in this manuscript by Ferber et al., and is computing the difference between the sponsor provided annotations minus the ones in the warehouse, and then looking at the Bland-Altman plotted slope. And what you can see here is that in the single ascending dose study, there was significant, what is called significant negative QT bias. In other words, that this data set, the QT measurements provided for long QTs were shorter than those calculated by the automatic algorithm in a consistent fashion. And we did a sensitivity analysis to see how the concentration QT relationship looked like when using fully automatic measurements. And in this case, which is the right-hand side panel, what we observed was that there was no difference in the slope. So in this particular case, we concluded that the data, so the automatic measurement suggested that this discrepancy was likely due to the difference in the automatic ECG methods used in the SAT study versus the thorough QT study. What else we do? We do a lot of regulatory science projects at the agency, and many of them have to leverage the ECG waveforms in the warehouse as well as the clinical data, and that allows us to advance the cardiac safety field. And here I'm enumerating a number of publications. There are others, and I include here the one recent publication by Cindy Green, who will talk after me, and what are the implications of this research. So let's move now into how to send data, or in other words, how to send annotated ECG waveforms to the agency. Again, I'm going to reiterate what is changing and what is not changing. So what is changing is the mechanism for uploading the ECG waveforms to the agency, and that's going to be moving from uploading data from the ecgwarehouse.com to upload the data directly through FDA electronic submissions gateway. And I include a link here also for your reference, even though the applicants that are submitting the clinical trial data will be familiar with this mechanism already. So there are no changes in FDA requirements regarding when ECG waveforms are required or not. And there are no changes on what is the file format for sending digital annotated ECG waveforms to the agency either. It continues to be HL7 annotated ECG standard. In this slide here, I want to outline how the data workflow looks like. The applicant may have collected the study information, have the clinical study report, tabulated data sets, and then the annotated ECG XML waveforms that might be handled by the applicant or by a CRO. But at any rate, what we end up receiving is we have two entry points as of today. One is through the electronic submission gateway on the right-hand side, where the applicant submits all the documentation and data sets following ECTD standard. ECTD stands for Electronic Common Technical Document, and it's a way to organize the clinical data. So XPT files containing QT measurements come through the gateway, but then the annotated ECG waveforms with those QT measurements actually come through the ecgwarehouse.com. So what happened behind the scenes that you don't see is then the interdisciplinary review team, when it gets assigned to review one of these trials or a set of trials, if they are part of a substitution request, will pull data sets, reports, and any other relevant information from what is called the electronic document room, which contains the folders of the trial following ECTD standard. And then we'll pull we also pull the ECG waveforms from the warehouse, and then we mirror and connect everything in-house, which allow us to do things like what I described before in terms of sensitivity analysis with different QT measurements. So what is changing again is that then we are asking now to submit the ECG waveforms not through the ecgwarehouse.com, but rather together with the rest of documentation in the ECTD folders. And this means that basically we want, or we recommend to include the ECG waveforms under a study, under the study folder, under miscellanea, and within a folder named ECG. So all the ECG waveforms can be included there when preparing your submission. And in that way, what we need to do is we're avoiding this two pathways approach where we have sometimes to keep things in sync, and this facilitates the process. So what is the impact then on how the ECG HL7 files flow from the applicant, right? So when submitting data through the ECGwarehouse.com, the ECG files, the XML files, are uploaded in a separate fashion from the clinical datasets, from the tabulated clinical datasets and study reports. And what the ECGwarehouse.com website provides is a validation report. And in some cases, the applicant or the CRO will work to figure out whether there is a format issue or what are the issues that may pop up in this validation report, and may or may not get support from the contractor that is currently running the service. This upload mechanism is scheduled to stop being available sometime this month. And although the warehouse itself will be kept up and running until later this year, but the upload will not be longer possible at the end of the month. So instead of, once you have the XML files ready, either if you re-adjudicated or whether they are containing automatic measurements, either way, what we are asking again to do is to include them and submit them together with the rest of the study data in ECTD format, so that it comes through the electronic submission gateway. In this case, the files are uploaded as part of the ECTD submission, so it's part of the folder hierarchy. We are releasing a set of ECG tools that were pushed this morning, so you can, I'll have a couple of links later on the next slide. And like with other data that is coming through the gateway, the support is coming directly from the electronic submissions gateway. In both cases, any clarification regarding the data itself or the submission can be sent through the review division, so the appropriate CDER clinical review division, and they will relay the questions to the IRT or the appropriate team in the agency to address them. So I mentioned in the prior slide some open source annotated ECG tools, and we just released a couple of tools. One is a library and command line interface that allows you to generate pretty much indices and logs of whether or not the ECGs are as expected. And we're also releasing a graphical user interface to even facilitate that further. The user interface in particular is relatively simple because the goal of this tool is really to be able to generate an index that you can actually check against and not to replace any other tools that are out there. And with this, you could generate indices in the same way that we do when we are cross-referencing with the raw data sets. You can take a look at your measurements. You can take a look at the XML, and you can see your subject listing in a user-friendly fashion. And with this, I'm gonna pretty much start stopping here and with this summary. Again, there are no changes in the FDA requirements for ECGs. What is changing is that the upload of ECG waveforms is moving from ECGwarehouse.com to the FDA electronic submissions gateway, that the HL7A ECG format continues to be the format for submitting waveforms. And if you have any questions regarding a particular submission, please contact the project manager in the appropriate CDER review division. And for any other information about clinical cardiac safety studies, you can visit the interdisciplinary review team, which again, I included the link here. And I'm looking forward to the discussion later on. Thank you so much for your attention.
Great. Next we have Dr. Cindy Green from Duke Clinical Research Institute, who will be presenting about the CSRC ECG warehouse.
Okay, good afternoon. Can everyone see my screen okay?
Yes, we can. Thanks, Cindy.
Awesome, thank you. Hi, I'm Cindy Green and I work at Duke University as a biostatistician by trade. And one of my responsibilities is the management of the CSRC ECG data that we store here at Duke. My slides won't advance. There we go. So I just want to very briefly give a background of the CSRC ECG warehouse, the data that we have currently in the CSRC ECG warehouse, some examples of completed projects that we have done in the recent years using that data, the process for requesting data if you have a project that you're interested in pursuing, some future research ideas, and hopefully plans to expand the ECG warehouse. So very briefly, this all started to want to study more in detail the QT interval. So the QT interval is just the time from the start of ventricular depolarization to the end of ventricular repolarization. And as we all know, this prolonged repolarization can cause ventricular arrhythmias, which can be fatal if left untreated and sometimes can come up without warning. So in the early 90s, there were a lot of cases of this Trossard de Pont reported in post-marketing studies, which led to several study drugs being recalled. These from 1991 to 2003 were approved for use and then subsequently recalled after QT prolongation was detected. So in May 2005, the ICH E14 guidance document was developed, which requested all sponsors submitting a new drug application to conduct what's called a TQT or thorough QT study. And this was to characterize the QT effects of the drug as related to a drug concentration. And these studies primarily enrolled healthy volunteers as opposed to individuals who may be at increased risk of these arrhythmias. And since 2006, more than 500 of these studies have been performed. And in conjunction with the FDA, a QT interdisciplinary review team was put together to review data, and it was comprised of pharmacologists, cardiologists, statisticians, etc., to review these data. And one of the resulting factors from this was the establishment of the warehouse for ECG data in 2004, just as Jose has been describing for us. And this was a centralized repository for ECG submitting with these thorough QT studies. And it provided a direct upload of annotated ECG waveform data using HL7 ECG standards. And now, as we know that, Jose, I'm using probably a later slide, there was 10 million ECGs at the time that I was perusing these data. Now there's more than 12 million. And also in 2012, continuous ECG data from 24-hour Holters have been collected. And at the time, there were more than 18 studies and more than 2,000 individual recordings that I hope that we can tap into from the CSRC side. So the majority of the rest of this talk will talk about the ECG data that we have at Duke for research. And in 2006, a memo of understanding between Duke and the FDA was created for the CSRC. And within, as part of this charge for this consortium, was to tap into this larger FDA Mortara ECG warehouse that was established in 2004. And what we did was go to sponsors and ask if we could have permission to take data related to non-drug, so baseline or moxifloxacin data, nothing pertaining to the actual drug given during the third QT study. And we did receive permission from sponsors and acquired 20 third QT studies. We've also collected more than 500 congenital long QT ECGs, and these are available for research projects approved by our scientific oversight committee. So like I said, we have 20 third QT study data sets, of which are stored within the CSRC. These are various sizes based on number of observations, number of ECGs, and the number of subjects. The design can be crossover or parallel, so maybe every subject got all superdose, moxifloxacin, and or they could be parallel. So maybe subjects only got one dose of drug and are compared to other subjects who didn't get the drug. The baseline can be pre-dose, it can be time matched. And like I said, we have around 1,400 subjects, over 100,000 ECGs, and these are healthy volunteers, so the average age is around 35, and it's about half male, half female. So what the CSRC ECG Data Warehouse Committee did was put together a process for how these data would be used. And we separated the data in our warehouse into what we called algorithm development data sets. And these would be data sets that we would share with interested investigators for further exploring their research and possibly developing their algorithms. And we call these development because they're unblinded. So we send the ECGs along with some limited baseline descriptors, such as the age and the gender and what treatment arm they were in, but only for the baseline placebo and moxifloxacin data. At that point, if the investigator is interested in pursuing further testing of their data, we will do a blinded comparison for them where they actually only generate the data based on their ECG algorithm and send that to the CSRC for a blinded comparison. And we published with Dr. Clickfield in 2010 the rules for how to go about obtaining data and the rules for those use. And then in 2012, we published our first project where an algorithm was tested against 2 third QT data sets in our warehouse to determine if their algorithm, how it was performing. What we were able to do using both a parallel designed and a crossover designed third QT study was show that the baseline unadjusted QT differences were less than a millisecond different in 7 of the 11 time points of which ECGs were collected. And also the 90% confidence intervals were almost identical in width, if not smaller, for the crossover study. That led us to another automated algorithm project in which the first project, we had 4 vendors who were interested in just seeing if there were systematic differences between ECG algorithms in practice. And what we did was put together the 4 interested parties in one room during a conference to take blinded interval measurements, which were done and in the normal moxifloxacin as well as long QT groups. This was successful and was followed up with another project because there were additional vendors that were interested in participating. And we actually redid this blinded measurement with 7 algorithms. And what we found were clinically small but statistically significant group differences do exist in interval measurements between algorithms. The purpose of this wasn't to determine if one algorithm was better than the other, but to determine if there were systematic differences. And these results were published in 2014 in the American Heart Journal, as well as the second study in 2018. These same data sets led to a third project. One of the researchers noticed that there were sex differences on some of the ventricular repolarization durations, and that there were differences in type 1, 2, and 3 long QT syndrome patients, particularly in the congenital type 2 long QT syndromes. So this further led to another publication in the Journal of Electrocardiology in 2020. So a third publication using these same data, which was exciting for our group. Currently, we are looking at another project with these same data, much as what Jose talked about in development bias and sensitivity analyses. This would be related to assay sensitivity in third QT, and if we could remove the need to have a moxifloxacin treatment arm. We have received approval from our vendors to use their data for this project, and hopefully we will be pursuing that this year. Another project in our women's initiative is QT heart rate corrections. So looking at gender specific, whether it be population corrections or Fredericia, is that which population, which QT correction does better in which population. And then as well as what Celine will talk about in collecting the sudden death cardiac in the young data warehouse. So just a little bit about how, if you want to use data, how you can go about it. There are 5 steps to receive ECG data. The first, just contact us. Email the cardiacsafety@duke.edu and let us know that you're interested in obtaining data and provide a couple sentences describing the project. Once we get that initial email, we'll send you a project submission form for you to send us some further information and more details about the project. And then we will forward it to our scientific oversight committee for review. If they have questions, they may do these questions through email, or they may ask you to attend a call to further discuss your project. If it's approved, I will be putting together the data for you and sending it using a secure method. These would be the algorithm development data, so they would be unblinded with the treatment assignments and other baseline data. And if the results are promising, we would ask that you share your initial research with us and then determine if you want to pursue algorithm testing with the blinded data. So the response to proarrhythmic risk increase worked well, but there's still a lot of work that we need to do in developing and assessing new methodology. And one of those charges, we would like to expand the CSRC ECG warehouse by collecting other thorough QT studies, perhaps in such as the pediatric population or other populations of interest, and particularly continuous ECG recording, so we can do other testing besides static ECG comparisons. So to help pursue that goal, one idea was to add a request to this new website for submitting the data to ask if you would be willing to share data. And it would just be a simple yes or no if you'd be willing to share the placebo arm and moxifloxacin arm data only, no patient specific information, no drug type information of that kind, just if you would share the baseline data. Doesn't mean that we'll tap in and use it. It just means if, you know, if somebody's interested in using the data and we need to grow the warehouse, we would be able to tap that data without asking. As we all know, this multiplies the scientific contribution of the original research by promoting any new research we can with these data. I'd like to thank the CSRC Data Warehouse Committee for all their help in putting these data together and their continued support, as well as our friends at the FDA. And I will stop there. If there's any questions or more information, please contact me directly or send an email to the CSRC. Thank you.
Our next speaker is Dr. Salim Idris.
Hello, everybody. I'm aware of time, so I'm going to get right to my talk and stuff. Val, can we see everything?
Yes, you're good.
Okay, great. Thank you. So it's a pleasure to be here. I feel like I am piggybacking on a great thing. The past 2 talks have outlined the amazing work on the, what I will call the adult side of electrocardiography and the ECG warehouse. So what I'm going to talk about is getting into the pediatric side of things. I'm the head of pediatric cardiac electrophysiology at Duke and the co-director of one of the co-director of our heart centers here, but I also work directly with the CSRC. And I'm going to talk briefly about preventing sudden cardiac death in the young and what we're doing to develop a national pediatric cardiac screening data warehouse. I think it's very important that we put faces on this because we're not talking about clinical trials with medication. We're talking about sudden death in children. And this is just one. This is a person, young teen, and she died suddenly during a cross-country race here in North Carolina, but this is one of many. And in fact, hang on one second. In fact, the faces of sudden cardiac arrest in the young and sudden cardiac death are numerous and ones that you don't see. It's much more often and much more than's actually out there. And also to dispel some other thoughts and what's out there publicly, that this is about adolescence. This is a tracing of a 4-year-old who had a sudden cardiac arrest while playing in the park. And this tracing is from a defibrillator showing rescue shock that was given mid-tracing there with success. The other thing to dispel is that sudden cardiac arrest and sudden cardiac death is only about athletes and athletic competition, which it is not. This is one paper which showing, at least in Minnesota in one area, looking at a 14-year time span, that there's a 3-to-1 ratio of sudden cardiac arrests proportionally in non-athletes to athletes. And this is actually what we see in the hospital. It's not what's shown on the news. We see the non-athletes and the sedentary children that have had a cardiac arrest. So really, sudden cardiac death in the young is a national and worldwide public health problem, but it's a public health problem that suffers from inadequate reporting and documentation of events, because sudden cardiac death occurs in sedentary children, recreational sports children, and competitive youths as well. And it's at all ages, infants, children, teens, and young adults. Now, what we're working towards, and has been going on for obviously not just me, but many years, is what about prevention of sudden cardiac death in the young? Well, we do know what happens to athletes because athletes are studied, and we do know the diseases that have been attributed to causing sudden cardiac arrest and sudden cardiac death. It's a fairly consistent pie chart of proportion here. But if we look at those causes and we say what are potentially detectable ahead of time by history and physical, by electrocardiography, by echocardiography, and a combination thereof, you see these. So the issue here is, how do we perform primary prevention? And primary prevention is this: if we know who may be at risk, then we can perform an individualized assessment of that risk. We can apply strategies to try to prevent death or cardiac arrest. And those include things such as, medication, lifestyle changes, defibrillator implants, and such. But how do we do that? So the way we do that for the most part is screening. And that's through many things: history, family history, the patient's history, symptoms, physical exam, electrocardiography, echocardiography. Now, we know that screening is detection. Detection is pulling somebody out of the line that fails a screening test or tests, and that you do further tests on. And that has its fallbacks. If we had perfect screening, we would have this. We would have a step function that you have some combination of metrics on the x-axis that includes all of those things. And then at some cut point, threshold, you have a change that you can say somebody's at risk. But that's actually not reality, obviously. We're humans, and we all live on a probability curve. And this is a sigmoid or dose response curve that many of you know. And you can see here that the probability of disease or sudden death or sudden arrest will vary. The curve on the right is, for example, an 8-year-old male who's African-American. You have on the left a 14-year-old Caucasian male who's taking stimulant medication. Or subject 2 in the middle there is a 15-year-old African-American female. So these curves are varied, but we don't know how to detect that or determine where they are. So you end up with basically overlapping thresholds for somebody having a risk of some type of disease or sudden cardiac arrest or death. So knowing the pitfalls and the knowns, cost and efficacy and accuracy, false negatives and false positives, we also have to know that there are gaps. And so the gaps in screening are what we're trying to address. And knowing things such as what population to screen. And as I said, many in what's out there would screen only athletes or certain demographics, but we don't know that exactly. Measurement variability is a distinct gap. And as you all know, with your interest in electrocardiography, the ECG and all the tools and what Cindy talked about with what are the best correction factors, those will all vary in children quite a bit by demographic, things that we don't really know right now. There are limited data on the ECG that are normal data by demographics, by race, gender, ethnicity in the pediatric age group. And of course, there's also a lack of consensus on what's an acceptable false positive or false negative rate in this screening and in the population. So what we know right now is that cardiac screening in the United States does occur. It's occurring mostly by public stakeholders, parent groups and foundations, and some academic groups as well. It has been occurring regardless of academia and the medical community questioning efficacy, and regardless of lack of government or National Medical Society oversight and support. And it's also occurring regardless of a lack of a uniform approach. So the consequences of that are the populations being screened without the knowledge of short or long-term efficacy of that screening itself, and that data are being collected non-uniformly and stored randomly on paper or in electronic format, or not stored at all. And ECGs are being interpreted without normative data that are specific to the race, gender, age, weight, medication use, or other population variables that are part of that population and demographic. So through the CSRC, we started an initiative towards trying to understand this issue and make ourselves better. And we are currently in the phase of having the National Screening Data Warehouse pilot study, which is really regulatory, industry, academic, and public collaboration. So through the CSRC, with FDA support and funding through Duke, as well as our Parent Heart Watch, our public group overseeing many of the foundations and groups for screening, as well as industry support, as you see here. We are in the throes of the pilot study right now. So we have public partners that are extremely dedicated from these sites across the nation that give us a good sampling of what's out there, the differences in screening methods and metrics. And this has been a really amazing investigation in trying to develop a way to have uniform data collection and high quality data acquisition. So really the central goals of this are partnership, research quality data collection, establishing a sustainable data warehouse. This cannot be just a one and done project or goal. Reducing variation that we know is there. Evaluation of the ECG and screening data to determine effectiveness and advance the field is important and critical. Obtaining national data so we can understand our demographic, and also tracking screened individuals over time is critical to understanding effectiveness. So what we are doing in this pilot study is enabling electronic collection of a core set of screening information that includes such subject demographics, history and physical elements that are really common right now, medications or medication subtypes, and high quality digital ECG data being the signal data, not just images. And enabling follow-up of subject status, and then enabling centralized data collection and analysis. So the national pilot study has a data flow chart that looks like this. We have data collection that's up front. So this is out front. This is in the public. This is in the field on site that, as you can see on the left, the public are being accessed through our screening partners. Electronic information and paper information is being obtained, and ECG machines vary in vendor and type. And these data are then funneled through, uploaded through a web front end that we've developed through Box and using that. Then in a temporary server situation, that's where we take over and make sure that we can abstract the data as well as de-identify, and then perform the conversion of the ECGs to the FDA HL7 annotated ECG format, because we want to develop parallel tools and parallel data structure with what you just heard. Data storage then is in a national data warehouse once we have it in that common format. And then we can enable data analysis with the goal of having quality metrics and summary statistics being fed back to the screening groups themselves as a part of a quality improvement cycle. And then outcomes data collection is critical, and we are enabling that in this pilot project to interact with the public and interact with screened individuals over time. So just to go through a few elements of this, as far as the data goes in terms of the demographics and the history and physical forms, screening outcomes, So our problem was that, many groups have paper intake only in varied amounts. So we created a data dictionary and then also developed scannable forms on paper, because enabling each group with varied amounts of support to get to a fully electronic intake can't happen at this moment. So we're using multi-page forms. We use a software through a company called Autodata, which allows us to create scannable forms. And then we can scan those forms to image format using a standard scanner, either on site at the screening itself or after return from there as a centralized scanning, and then use software post-processing of the images of the forms to get these into the database. So here's an example of the form, or first 2 pages of the form. On the left is the first page, where you can see we have an individual participant fill out sort of the Scantron bubble fill-in format. The barcodes are actually a UID that will be tracked all the way through and used along with the ECG to make sure that we have all of these data and the ECG data in register in the future. On the right is the second page, which has data that we are including with COVID questions, as well as other common questions related to heart problems and other things that are known for questioning on prevention of sudden cardiac arrest and assessment. In these forms, we also have staff use forms, which will be physical exam findings to track and record, as well as on the right, which is an ECG outcome form, because these ECGs are being read contemporaneously on site or slightly somewhat after that. So we want to know who read the ECG that was being sent in, as well as what was the determination of that. Was it an abnormal or a normal? So then we on the back end can go ahead and do quality analysis and start to determine if there's variance between groups, between demographics and such. And there are difficulties with scannable forms and any paper forms. As you can see here, this is from a screening that we've done as a test screening with an individual who had difficulty writing block letters, answering one thing and then putting another thing. So when you start to scan these in and try to automate this process, there's clearly a lot of quality issues in curating the data into the database. But we do have a demonstration here from one on-site test of 161 individuals, which shows that we're able to abstract from these forms participant ages and gender, as well as ethnicity and race demographics for the proportions that were screened on that particular day, as well as measured weight and systolic blood pressures that were obtained, as well as a comparison of measured and reported heights from the individuals. So we have pretty good correlation here related to measured height and reported height as a demonstration that the individuals can fill these forms out and actually do some degree of answering questions on site. Another problem that we are having, that's not we're having, but we're in the process of solving, relates to the digital ECGs. Each group uses a vendor or ECG machine of their choosing. And of course, these various ECG devices have various file formats and outputs. So we are developing and developed the file transfer for lay users on site to be able to upload to Box to just a file structure to get the digital files up to us. And then we have developed conversion software with help for changing the ECGs into the FDA ECG, annotated ECG HL7 format. And I just want to thank Cardiac Insight, who's been at the table all along with this, for their support, as well as AMPS, who's been a key participant and collaborator with helping us with these conversions. And also we're developing new data software, as well as using current ECG signal analysis software, and want to do this in parallel with everything that Jose has talked to you about. So COVID was a problem, along with everybody else having COVID problem, and screening stopped because this is a public event. But because of the retrospectively collected ECGs by the groups, we actually have so far have over 10,000 ECGs in our warehouse that we've been able to do our test runs on and start to work with the data on that. And it demonstrates proof of concept for what we're doing. So this is just an example of some of the waveforms. This is waveforms created, pulled out by software that I've written, as well as showing that we can do some degree of signal analysis on the back end to start doing annotations of these data and start doing some analysis in the future. And then pulling out the data that were auto measured from those devices. This is again just a demonstration of preliminary pilot and that we had over 9,500 ECGs that we could analyze and obtain age demographics and heart rates and things like that. But you can see here, just as a demonstration, pulling those automated results out of those uploaded ECGs, clustering of QT versus RR, as you would expect this relationship for males and females, and then able to do some degree of statistical analysis here, demonstrating that in this cohort, there's differences between males and females of the ECGs. And these generally are adolescents, as we would expect a male-female difference as such you see here. And just to validate this further as we get towards the end here, this was a recent article that came out that was to try to demonstrate and show standardization of ECG findings in children and young adults. This was from 2 hospitals though, so these were hospital-based ECGs. Over a long period of time, they had 27,000 ECGs. But if we look at the data that they collected over different ages, we collected in this red box. And if we look at the 2 data sets, on the left is from that paper, they collected 9,300 ECGs over that time period, and we have 9,500. And we're showing similarities of QT based on gender differences, of course, in this age group. So what this is really just a showing that we have feasibility of acquiring meaningful, large data sets from the broad public demographic across the US with the mechanism that we're doing right now. So what we're on our way to doing here is from pilot study to really a national pediatric cardiac data warehouse. And just to put this in graphical form as a future state for this healthcare resource, we have the data in the middle in the warehouse, as are the screening groups that are submitting data and where we're acquiring data. I is industry, and A are academic institutions, and F is the FDA. All of us interacting together with this data set. But what we can really achieve that is where we all work together, so we can analyze this data to try to get a better understanding of sudden cardiac death in the young. We can then move these out to the screening groups and improve screening efforts, improve understanding, improve uniformity and quality of data collection. And then in the end, what that means for you all in terms of pharma and the device industry is that we will have a high quality data set that we are able to curate in the pediatric space and understand the differences across that demographic and provide advances. So there are many stakeholders here. The public are clearly a stakeholder, sports organizations, and those in that space who are trying to prevent sudden cardiac arrests in the youth. Academia clearly has a stake in this, sports medicine groups and all of our academic societies, medical societies, including one I'm mostly associated with, which is the Pediatric Congenital Electrophysiology Society. And then industry in terms of all of you and device manufacturers and ECG companies, we want and need normative data across demographics to move the needle on pediatric device development and pharmaceutical development, of course. And then from a regulatory aspect, this is indeed a public health problem. It affects all of the demographic of the US and worldwide. So pediatric population cardiac safety data is essential to have. So to conclude on this, sudden cardiac death in the young is a tragedy. It is a public health issue. Screening for diseases that cause sudden cardiac death in the young is primarily driven by the public and with minimal guidance or alignment of best practices across the US. And our current initiative is to enable organized public screening to generate high quality data in order to determine the efficacy of screening and develop data-driven standardization of best practices. And above all, we're establishing a national screening data warehouse, and it's a critical step towards the future that affects many key stakeholders and can drive improvements to prevent sudden cardiac death in the young. So thank you very much, and looking forward to your questions.
Thank you all. I realize that we're a few minutes behind over our time, but we do have a couple of questions that I think we would like to go ahead and address. And I would like Dr. Vicente, we have a couple of questions, and if you all will stay on, he can address those. The first question is related to submitting continuous ECGs through the warehouse. How is that done, or can it be done?
So it can be done in the same way that it's been done for the ECGwarehouse.com. Now, what I would recommend to do is depending on the total size of the specific submission, to check out with the electronic submission gateway folks to see what are the file limits or whether they recommend a different approach to electronic.
Okay, next question. Will it be possible for a CRO to submit an ECG through the FDA gateway on behalf of a sponsor? And how could this be done?
Right. So we have follow up with our colleagues in the electronic submission gateway and e-submission and e-data teams about this question, because this is a question that we are getting already. And the short answer is it's doable, but the long answer is not recommended. And the reason is the following. One has to so there are two phases really. One CRO submitting on behalf of just one sponsor, and then one CRO submitting on behalf of multiple sponsors. And there are restrictions that allows you to submit an authorization from the application owner to be able to do this. But there are some nuances regarding the synchronization of the ECTD data structure between the different submission submitters in this case, right? So again, what I would encourage you to do is to follow up with our ESUB team and check what the approaches would be. But I think the recommendation that they are gonna provide to you is that the owner of the applicant would be the one submitting the XML files together with the rest of the application to avoid synchronization issues in terms of sequence number and directory structure in the ECTD tools that you may be using. Regarding synchronizing the gateways from FDA with the applicant, as well as with any other CRO or vendor that are submitted on their behalf. That's the long story short. And I do have another question that came through the chat regarding what are the studies that are required to submit ECG waveforms. And again, the long story short for the ECG waveforms is check with your regulatory review division on whether or not they want to see the ECG waveforms. So the requirements for submitting have not changed. So if you have a study and you don't know whether or not you should send the ECG waveforms, please check with your regulatory review division.
Sounds good. I also would like to encourage everyone, for those of you who are not familiar with the CSRC, please reach out to me or anyone else that's our CSRC email address to find out about how you can join the CSRC, how you can get involved with any of our initiatives. We are continuing to grow, move things forward. One of the questions that came through on the pediatric side was, have we thought about collecting data from wearable devices?
Yeah, I think at the time right now, we have thought about that. It's obviously the future state. We have to prove first that the lead information that we acquire from a wearable, be it a single lead or a multi-lead device, is going to be adequate for differentiating the diseases that we're looking for with sudden cardiac arrest. So we have to start with the gold standard 12 lead at the moment. But then as soon as we're able to pare down what's a limited lead set, if we can do that, that would be what we would do. But of course, we need the data into the ECG pediatric warehouse to be able to do that differentiation, to start to determine if some leads aren't necessary for that differentiation of the diseases.
Sounds good. Well, if there are no more questions or comments, we would like to thank you all for your participation. If you have any questions or that you think about later, please reach out to us. And as a reminder, we will have this posted on the CSRC website as well as our YouTube page. Thank you all once again. I do want to do a brief commercial. If you're in the Shock area, the CSRC will be conducting a think tank with CRT on March the 19th, and we invite you all to participate. We look forward to talking to you soon. Thank you.
Thank you, everyone.
Bye. Bye-bye.
Bye. Thanks, everyone.
Thank you and bye-bye.